Thank you. Thank you. Thank you. I must admit, when Professor McBride invited me, I was a little reluctant because during graduate school, one of my toughest job was to explain what I do, because nobody can relate to what I do. It's specialized, it's molecular, and in order for someone to relate to it, you have to put something that it's common. And I'm not saying that what I do is something so special or grandeur or whatever it is. It's just different. It's hard to relate. And so when I moved to Houston, and I work now in the cell and gene and I worked at the Cell and Gene Therapy Center, and I did a cancer-related project, I was relieved because I can say, I work on cancer. And everybody's going to say, oh yeah, find cure in cancer. It means nothing, because you're never going to find cure for cancer, because cancer is such a massive disease with hundreds of thousands of different variations of it that no one's going to ever find a cure for cancer. I don't know whether that's a news for you or not, but it's not going to happen. We may find cure for certain types of cancer, but not to the disease of cancer, because it's so massive. I worked with T cells, acute lymphoblastic leukemia model, which is one small version of leukemias, which are small versions of all cancers that are known to men. But I could say that, and I can relate, and that will end it there, of course. Nothing changed of what I did, but in graduate school, I was working with protein folding. What? Protein folding, what's that? I had a really hard time explaining what I do and why I'm spending money, and when people pay me, as a matter of fact. But I'm trying, I will try here to relate as much as I can. And let me demonstrate why I was worried as well. So at the end of my graduate work, actually not quite at the end, but about third year, we made a significant discovery, which I'm going to tell you about, a little bit about here. And the local newspaper wanted to come and interview us. And I was a little nervous, and then I saw an interview that they did with my mentor, and the stuff that he said. And I got really worried when I read what he said, because I'm like, we're not doing that. When did we do heart-related research? When did we do research related to addictions? When did we do behavior-related research? I'm like, I never did that. I work in the lab. I work with protein called phosphorylation protein. That's it. That's all I knew. And he was saying all these stuff that it just amazed me, and it worried me so much that I had to go and read up a little bit so that I will be able to explain what I do to an interviewer, because I couldn't tell him exactly what I do, so that I can relate to people. But since then, things have changed. So hopefully, I'll do a little bit of a better job. So today, I wanted to start a little bit in a way that can relate, and it can explain what does a scientist do? What do we do as scientists? And why do we sometimes perhaps appear a little quirky, or kind of walk around where we don't really notice people, or we're still normal, but we just think we're quirky. I don't know, something like that. So I decided to start with this picture. And everybody who sees this picture says, wow, this is a beautiful female eye. I don't know whether you could tell that it's female, but it is a female eye. And we all know what it is, right? You can all say, yeah, this is an eye. And if I ask you, so what's the function of, what does this organ do? And you will say, well, I mean, that's an organ that allows you to see. And that's pretty much what you know, I would imagine, unless you know a little bit more about the anatomy. But it's an organ that allows you to see. You're using it to see now. But a scientist doesn't end here and say, okay, well, let's find out why. Let's be a 2-year-old now. But why? Why can an eye see? What makes it so special? And then if you're bold enough, or you open a few pictures here and there, you will find out that an eye has a certain structure, and that the eye has, the sclera that surrounds the eye, that the eye is connected with an optic nerve, that there is a spot here in the eye called macula that really allows for colored vision. Retina is the real organ that perhaps allows you to see, maybe not. There is the lens here that focuses light. Here's the iris that makes eyes so beautiful, either blue or brown or green or whatever they are. But in anatomy, it's just simple organ that regulates how much light will come in. That's basically the function of the iris. We have the cornea, and the whole eye is filled with this gelatinous matter that is so clear and allows all the light to go through without any interference. And you'll say, okay, wow, we finally understood why we can see. Yeah, but why can this eye see? Why can't the ear see? Why can't my hand see light? What makes this organ so special? And then you further think, okay, well, histological analysis shows that there is a special organ as part of the eye called retina, and there is a spot in that retina that allows you for you to see color and allows you to see focused objects. And then, but then again, what makes that part of the eye? Why can't the sclera right outside, the connective tissue, why can't that tissue perceive light? What makes that tissue so special, the retina and the macula? And then you do further analysis, and then you take that little spot over there, you cut it with a microtome, and very thin slices, almost so thin that you can see through them, put them on the microscope, color them with different dyes, and then you say, wow, look at that, it's a whole world out there. I mean, there's different layers, different colors, some are dots, some are lines. What's going on? And then at some point you realize, or you learn, that there are certain parts of these layers, like for example right here, the rods and cones, that are actually responsible for the light perception. It's not just the retina itself, these special cells, and we call those cells photoreceptor cells. And then you say, okay, photoreceptor cells are the reason why we can see. And then you say, but why? What makes these cells so special? Why can't a brain cell see? Why can't the liver cell see? Why does this cell can see or perceive light? So the photoreceptor cells are these very interesting cells that look like rods or cones right here. You can see them. And you say, oh, so look at them. They look a little bit different than the rest of the cells. But what again? What is it making them so special? What's that? And then further investigations shows that these portions right here that you see as rods are actually these gigantic, for a microscope, gigantic layers of membranes that are just stacked on top of each other, hundreds of them for each one of them. And it makes these cells look a little different. They also have a nucleus, they also have other organelles, but there's these portions here that are different. And if you take them out, you can't see anymore. So they have to be there. And then you further investigate them, and you find out in the better sections that, look at them, there are hundreds, tens of hundreds of membranes that are stacked. And you're like, but what makes these membranes so special that your eye now can see? What's the big deal about them? And then you start investigating, do more experiments, do more studies, and try to figure out what's happening. Why is this so special? And you find out that there is this protein in these membranes, and that protein is called rhodopsin. And you can see the rhodopsin in this case was stained with a red stain right here. That is in the rod outer segment parts, basically in these portions of the cells. And these portions of the cells are rich on these protein called rhodopsin. And if you don't have the rhodopsin, you can't see, even if you have the membranes, even if you have the photoreceptor cells. If you don't have rhodopsin, you can't see. And you start wondering, why is that? What makes that protein so special that it can see that allows us to see this presentation or light in general. And then further investigation, you find out that rhodopsin is this protein that is made out of these helices, 7 helices that span the membrane. Yeah, but there are about hundreds of them, of those kinds of proteins in the body. You smell because of the similar proteins like that are in your nose. You taste because of very similar proteins that are in your tongue. And you wonder, why that? By the way, your heart races faster because of proteins like that in your heart. Adrenaline signals through a receptor like that as well. And I was like, what makes this protein so special? What is it? Notice a pattern here. The question why, the question what. That's a very common question that we always ask ourselves. You know, why is this the way it is? You never cease to wonder. So we keep wondering, why is that? What makes this protein so special that among hundreds of others, it will allow us to see light? You don't have it, you don't see. So further studies show that this protein can change with light. If a light shines on it, it changes. I mean, I hope you can see that there are some slight changes of this protein compared to this protein. It's a diagram, but you can see that some of these helices, in this case, are slightly different. They're twisted a little bit differently. And you start wondering, how can light change these proteins? Why would light do that? Why light doesn't do the same thing for our skin, for example, or for your nails? Why doesn't it do the same thing? What's making this protein respond to light? And further investigation showed that there is this small molecule right here, hidden. And that molecule is called retinol, changes with light. So normally, during dark, the retinol structure is the following. But when a photon of light reaches the molecule changes from an orientation called cis to trans. Basically, there is a rotation. I'm trying to find a good position here. There's a rotation around this bond. All it does is switches like that. That's all it does. And that's caused by light. Now, if we don't understand the structure of this molecule, if we don't understand how we gather this information, or how do we determine that structure, we will not be able to answer that question. We will not know why we can see. So suddenly, chemistry that you learn in general chemistry or chemistry that you learn in organic chemistry becomes very relevant for this question. The reason why we see light is because this molecule called retinol rotates as a result of a single photon of light. It hits it and it rotates. And it causes now the receptor to change its shape. And when it changes its shape, it interacts with these proteins and passes a signal. But how does that signal become a perception? It has to go to your brain somehow and tell you, you see. That's light. So there's a model for that. Okay, once the receptor rhodopsin reaches, or is reached by light, and the molecule switches, it triggers, it interacts with these protein called a G protein heterotrimer. It means that this is a GTP containing protein that has 3 different subunits, alpha, beta, and gamma. And when it interacts with them, it causes this protein to lose GDP, which is a nucleotide, and to be replaced by another nucleotide called GTP. It's just a matter of a phosphate here, just a matter of a phosphate. All right, after that, you activate alpha subunit, and the alpha subunit starts migrating very fast, by the way, on the membrane, and it interacts with a protein called phosphodiesterase. And when it interacts with the protein, phosphodiesterase protein, it causes the inhibitory subunit, or that part of that molecule, to be released. In other words, there is nothing now to stop that enzyme from working. And what does it do? It takes guanylyl monophosphate, it takes cyclic GMP and converts it to just GMP. It just changes the way the molecule appears. By doing that, it takes away cyclic GMP, which is required for this channel on the membrane, the photoreceptor cell, to close. Now, if cyclic GMP is there, the channel is open. Sodium and calcium flow inside the cell. But when you take away cyclic GMP, the channel now closes. And when the channel is closed, I don't know how many of you have taken physiology or been to a physiology or biology class, but something like hyperpolarization happens. I don't know whether you're familiar with that term, but hyperpolarization is basically changing of the membrane potential. When you change the membrane potential, you change the signal that that cell passes to the next cell and the next cell and the next cell so that you perceive light. So when you do that and you cause the hyperpolarization of the membrane, you're passing a signal down to the axon, to the neurons in the brain to tell them that there's something happened and I was closed, basically. And then the whole process goes back. There are mechanisms that neutralize all these effects that one photon of light caused, and then everything after that repeats again and again and again and again and again. So what have I told you so far? That light causes a molecule to rotate, another protein to change its shape, that binds to another protein that also changes shape, then it binds to another protein that also changes shape, that basically depleted cyclic GMP that is required for a channel in the membrane to close so that calcium and sodium can no longer go in, hyperpolarizes the membrane, a signal is passed to the neurons in the brain to tell you a photon of light was received. This is coming from one receptor. There are millions, and they fire all the time. You can imagine the amount of information that is going towards the brain for you to be able to see this picture at this second, and the next second, and the next second. This is a very complex process. We think we understand it. We're lying to ourselves, but we have some form of understanding. So why do they tell you all this story? I mean, first, to try to relate, to tell you how my work matters, or what I've done, and how does that matter to the world, I guess. So I'll show you a story, how we worked on a protein, this protein here, transducing beta and gamma, as part of the heterotrimer that is required for passing of the signal. I'll tell you a story, how did we find out, how does this protein express in the cell and becomes functional? Because if it's not functional, you can't see. And not only that you can't see, but 30% of your hormones will not be able to work if it doesn't work. You will not be able to taste anything, you won't be able to smell anything if this protein is not expressed properly. Just that fact. So how did we figure that out? So basically, my work could be described with this figure here, showing that when the G beta subunit mRNA is bound by the ribosome and translated into a protein, then that protein gets picked up by another protein called phosphorylation-like protein, which is phosphorylated, which is important, and then gets brought up to a massive complex called cytosolic chaperoning complex, which folds proteins that cannot normally fold themselves. Helps to be folded into a proper shape, then another subunit comes in, they're associated properly, and then delivered to the membrane where they will interact with the alpha subunit, forming this complex shown right here, the real structure. Okay, so how did we come up with all that explanation? Okay, the story starts a little bit farther away with this protein called phosphorylation-like protein. So I worked with that protein. And up to this point, we knew that phosphorylation-like protein is an inhibitor of G beta function, We had no idea that it's required for anything else, just that it inhibits it. It cannot function if it's present. So we thought, okay, let's see what would happen if we generate cells that don't have phosphorylation-like protein. What would happen with G beta? And we anticipated that the expression is going to go up. So we went and inhibited phosphorylation-like protein with a special tool called siRNA, and I can explain later, I don't want to go into too much detail. Lamin AC, we use it as a control to show that siRNA has specific effect. In other words, when we use siRNA against lamin AC, phosphorylation-like protein expression doesn't change. When we use phosphorylation-like protein siRNA, lamin AC doesn't change, meaning that we're not affecting other proteins in general, but specifically phosphorylation-like protein. And then what we saw is when about 85% of the protein, phosphorylation-like protein, is out or inhibited, and it doesn't exist. In other words, we've suppressed its expression. About 40% of the existent G beta gamma disappeared somehow, which was very interesting. We expected completely the opposite of this experiment. We didn't know how to explain that. We didn't know what were the reasons for that, but it was definitely a puzzle. So we said, why did that happen? What can we do to figure this out? We checked the mRNA, it was fine. The stability of the mRNA was fine. The genes seemed fine. It was something with translation of the DNA to a protein. So we said, let's study translation then. So this is what we did. We took cells, we treated them with siRNA. It's called RNA interference. Another word is that we knocked down the expression of phosphorylation-like protein. In other words, we decreased it to a minimum that we can get it. And then after we were sure that the phosphorylation-like protein is very low, we did an assay that's called pulse chase. So we took radioactive amino acid with radioactive serine, and we added it to the media where we grew these cells. By giving the radioactive amino acid, we can follow proteins that are synthesized from this moment on. Only freshly synthesized proteins. We basically say, we don't care what we have before, it doesn't matter, but we're going to look what happens from now on. Is translation affected or not? So we pulsed with radioactive methionine, and then we chased it after a certain time. We lysed the cells, and we wanted to see how much of the newly synthesized protein is present in the cells. So we pulsed the cells. 20 minutes later, we lysed and detected. 40 minutes, 70 minutes, 100 minutes. In the control, where we have perfectly fine phosphorylation-like protein expression, you see a nice increase of G beta and its partner G gamma. Here it is, a normal expression. But when we don't have phosphorylation-like protein, beta gamma doesn't seem to express. And we said, okay, so it is translation then. But what part of it? Or is it only translation? Is there something else that may be happening? So we didn't know. So we started investigating different portion of the protein. We were working on a side project trying to figure out the importance of what's called a post-translational modifications of the protein. In other words, there are certain signals, and signaling in cells is phosphorylation. I'm not sure how many of you have taken chemistry, but a phosphate is basically one phosphate connected with 4 oxygens. That's a phosphate group. And when that phosphate group is added to proteins, they get either turned on or turned off. In other words, that is a signal for them to change and signal further, or for them to change and stop signaling. So that's what phosphates do, usually. So we were working on what triggers phosphorylation-like protein to be active or inactive. And we basically took the protein, this is the sequence of the protein, These are all the different amino acids. And we basically shredded it into pieces. And we put it into an instrument called protein mass spectrometer, where we measure the masses of each of these fragments, trying to figure out what is different. And this is what the results look like. And by analyzing the results, we found out that there are 4 phosphates added to this protein. 1 is added here, and up to 3 are added here. And we said, oh, interesting. That means that these phosphates may or may not turn on this protein. Let's investigate if these phosphates have a role in the activity of this protein. So what do? We mutated. We went in, pulled the gene out, basically, when we had it ready. But we went and substituted all these amino acids, they're called serines, with alanines. And alanines do not get phosphorylated. So we did mutagenesis. We changed them all, and then we said, okay, let's see whether that's going to have any effect on the assembly. Okay, so we did a similar pulse chase experiment on a much bigger level now. And we have, this is the wild type, meaning the normal, natural phosphorylation-like protein. The 25 to 296, this is, I'll just use this one, it's a little bit better. Basically, these serines, the 25th position and 296, we replaced them with amino acids that do not get phosphorylated. Same for 1819, 1820, 1920, and then all 3, 18 through 20. And we wanted to see how will they affect the function of this protein. And would that have a role in the expression of G beta and gamma? So with the wild type, again, 20, 40, 70, 100. Wild type here, slowly increasing the way it's supposed to be, right here. So we said, great, we got a good positive control. The assay is working. Then we looked at the 25 to 96, as you can see, almost no change. It behaves just like the wild type. So phosphorylations at these 2 sites don't do anything. They are not important for this process. They might be important for something else, but not for this process. So we looked 1819, 1820, 1920, and they pretty much grouped into one area right here, showing that there is an effect. They do affect the expression of G beta gamma, but not significantly. However, when we take all 3 of them, 18, 19, and 20, the assembly and the expression of G beta gamma was significantly diminished, almost as if we don't have phosphorylation-like protein expressed in the cell. Okay, so that tells us that phosphorylation plays a major role. That's the signal for the function of this protein. So what else can we figure out? Then we said to ourselves, all right, let's see, is it really the expression of G beta, or is it the assembly of these 2 proteins? Because if those 2 proteins don't get assembled, beta and gamma, they are not functional. You can have plenty of each one of them, but if they're not together, they don't work. They cannot work. They're not functional. So we said, okay, we'll set up a few experiments with cells, and we'll express these different variants in different cells, and we'll see how they affect the association of phosphorylation-like protein with G beta, and G beta with phosphorylation-like protein, and G beta with G gamma. Let's see what we got from this. Okay, so we got the wild type. When we express the wild type, it pulls down G beta 1 fairly well. When we express the 18 through 20, we also pull down G beta gamma. Well, a little bit less, but still okay. When we express the one that we truncated, we replaced half the protein with another protein, that didn't change much. But when we removed the first 75 amino acids, the expression and the binding of phosphorylation-like protein to G beta was significantly decreased. So there's something in that section of that protein, which includes, by the way, the 3 phosphorylation sites, is telling us that it's very important. Then we looked, how does G gamma and G beta associate in the presence of the wild type, the real phosphorylation protein, or in the presence of some of these mutations, including this one. So what did we see? The wild type, this is the association after pulse chase, seems okay. That's the norm. This one is pretty much the same. However, when we don't have the phosphorylation sites, only beta, no gamma. When we remove the first 75 amino acids, only beta, no gamma. So beta gets expressed, but it cannot associate with gamma. And we set out to ourselves, what does that mean? What is really phosphorylation protein doing? It doesn't seem to be important for the expression, but it seemed to be important for the association. In other words, it has to play some kind of a role to bring protein B with protein G, basically. And we did a few studies afterwards that basically showed that phosphorylation protein was important for the release of the folded complex. Here you can see that gamma is expressed, but it's not associated in this case or in this case with G beta. It's there, but it cannot get together with beta. So something is preventing it. And that's the question that we answered, saying that phosphorylation protein facilitates the assembly of these proteins. So I'm going back to this original schematic. Here we have the nascent G beta, the newly synthesized G beta. Phosphorylation proteins helps it to associate with the chaperone that's going to fold it into a particular shape. It binds to it. Now the protein is folded into a particular shape. And then when the protein is phosphorylated, it releases G beta so that G gamma can bind on the opposite side. Once we have the heterodimer ready, then it gets delivered to the membrane where it associates with alpha, and now we're ready to function. Again, what's the significance of this? We figured out the natural way of expressing a protein that is responsible for you to be able to see, taste, smell, for half of or a third of your hormones actually to be able to function in your body. If this does not happen, well, you can imagine life without this. But it helps our understanding. And because of this, we were able to see more clearly, pun intended, how the body works. And it's a small question. We had no idea when we started this project that this is how it's going to end. It took us about 4 years to come up with this. And it took another 5 years later on as a follow-up to another graduate student to follow up. So it takes a tremendous amount of time to make a jump from here to here. It takes me, I used to say, it'll take me 5 minutes to tell you what you're going to need to do in graduate school. It'll take you 5 years to do it. That's the comparison usually in research. Now, why do we all do this? I really like this quote, and that's how I'm going to end. That there are two compelling reasons why society must support basic research. A lot of people don't like basic research, probably including myself to some extent, because it's a money, what's the good word to say? I didn't want to use a bad word for this one, but it siphons a lot of money, takes away a lot of money. You know, for me to be able to work on this project, at the time I was paid $17,000, but now it's $26,000 a year just to work on this project. I cannot tell you how much money I've spent for reagents. The operating cost of our lab in Baylor for the mice to be just housed was $10,000 a month, just for them to be housed, not to do experiments with them. Each experiment was about $2,000, just to do an experiment. So everything cost a lot of money. So why do we spend all this money? What's the benefit? One is substantial. The theoretical physics of yesterday is the nuclear defense of today. The obscure synthetic chemistry of yesterday is curing disease today. The other reason is cultural. The essence of our civilization is to explore and analyze the nature of man and his surroundings. And this is my favorite part because it connects science with religion. As proclaimed in the Bible of the book of Proverbs, wherever there is no vision, again, the people perish. So I hope this was somewhat entertaining to some extent, or revealing, or interesting. That's how I'm going to end. If you have any questions, I'll be happy to answer them. Thank you. Any questions?
You talked a little bit before. I think, when your mentor started talking about how this is connected with Alzheimer's and other kinds of things, would you like to? I can sort of imagine myself ways that it can be connected, but how do you connect it with some of these bigger issues that people are thinking about?
So like I was saying when I was showing my model, sometimes as scientists we're so focused into small details that we don't see the big picture. It's almost like you got to pull away from the whiteboard so you can see what you just wrote on the board and why that makes any sense. It's all guilt by association. I'm studying a protein that I know what it does. This protein, G beta, is responsible, for example, for you to be able to get angry, right? Because adrenaline works through an adrenergic receptor. This adrenergic receptor cannot operate if you don't have G beta. And you cannot have G beta if you don't have phosphorylation protein to be phosphorylated. Now the idea was like, perhaps this will open up venues or ways for some form of therapy maybe for certain diseases. Is that going to happen? Probably not. Because if you shut down this protein, you're going to shut down most of your body, the entire body, because it's so important. It's like a housekeeping gene. You don't have it, you don't live. Basically, that's the simple conclusion. The way I relate this to the world is we found that we basically understood how something works. And we were able to, following this work, to understand how the chaperonins work. And chaperonins fold about 10 to 15% of our proteins. Actin, for example, that supports our body, our cells, cannot be folded without this chaperone, but we don't know how. Phosphorylation protein is another one. Tubulin is another one. They all require this helper massive complex to work. By doing this work, we actually understood how this chaperonin works, and that each protein has its own helper molecule, like phosphorylation protein. And the phosphorylation protein helps other molecules to be folded so that our body functions properly. If it doesn't get folded, again, it doesn't work. And then either death or form of diseases. I haven't been able to relate this to any form of a disease, most likely because there is no disease that can be supported. Like I said, histamines work through G-alpha as well, G proteins. If you inhibit, for example, phosphorylation protein, you will not have beta. Histamine will not work. Some of you might say, good, I have allergies. I take pills to do that. Yes, but think of it this way. Histamine is a natural response to an allergen, something that potentially hurts you. By taking the antihistamine, you're actually fighting a natural process because you don't like to sneeze and the itch that you get. That's what you're doing. Aspirin, by taking it, you're suppressing a natural process. Most of the time, you shouldn't be taking aspirin when you have fever. You should let it go for 24 hours. Who wants to stay for 24 hours with 38 degrees Celsius fever or 101 degrees Celsius fever, Fahrenheit fever? No one. As soon as you get that fever, you're like, where's the Tylenol? Do you know, for example, that no one knows how Tylenol works? That's a surprise, right? There is no human on earth that knows how Tylenol works. We just know it does what it does. Would you like to know how it works? Yes.
I'm just curious about allergenosis. Were you saying it
Oh.
would be better not to die?
Oh no, that's not what I'm saying. I'm so sorry. I'm just saying that antihistamines battle natural process because we don't like the effect of it. That's what the antihistamines do. You're suppressing histamine, which makes you itch, sneeze, and all the stuff that happen during an allergic reaction. I am not speaking against antihistamines, please. I don't want my words to be taken that way. That's not what I'm trying to say. All I'm trying to say is that we're suppressing a natural process that has gone a little bit overboard. But we're trying to suppress it so that we can live a comfortable life. Otherwise, you're going to be coming here sneezing all the time. But that's how medicines work. I went to a conference, and almost every company I asked, it was for chemotherapeutics. None of them knew how the medications work. Most of the medications, we don't know how they work. We just know that they do what they do. Would you like to know how they work? Do you know how we find out how they work usually? By putting them on the market for 5 years and find out how they affect the human population. Yeah, you do clinical studies, but these clinical studies are very limited, right? Right, that's exactly what happens. Almost crippled a massive pharmaceutical company, Merck. It was a $2 billion settlement for, well, it wasn't, for Vioxx, which was a replacement for aspirin. It came out, people started dying because of it. So the lawsuit happened and almost crippled the company. Because you can only do so much in a clinical study. You take a certain population of people. I used to do this with my Chemistry 100 class. I would ask, okay, the class, design a clinical study. Who are you going to include in the clinical study for this new drug? Who are we going to include? Students? That's what we usually include, you know. Adolescent, 18 to 25. Those are usually the ones that are included, right? Are you including children in that? Is anybody willing to donate children for drug studies? Nobody wants to do that. Are we including elderly? Nobody includes that. Are we including people with certain diseases to see how they will affect a sick organism? We don't include that either. Now, we try to balance it to be equal male and female, because these two organisms are different. Not to mention cultural. Are we including white? Are we including African Americans? Are we including Asians? Because sometimes they react, people from different cultures react differently to drugs. So they are very limited. All of these studies are very limited. And a lot of times, we just do our best, and then we put it on the market, and suddenly we find out that this 1.12% or whatever is reacting badly, and they could die. So if I can give you any advice, if you know that the medicine is new, and there is another way, don't take it. That's the best way I could say it. Just don't take it for about 5 to 10 years until it's figured out what's going on with it. If it's brand new, avoid it. If you can. Sometimes you can't avoid it. People look for experimental drugs because that's the only hope that they have. And it's sad. So what I do hopefully explains natural processes. Maybe it won't contribute to therapy, but it explains a natural phenomenon. Understanding how the norm works sometimes help us see what's wrong if something goes wrong. So knowing the norm helps us understand pathology or how disease work. Any other questions? If there's time. I don't know whether there's not time, but Lachlan?
So you said that when you have like a 30,
Yeah.
the first thing people look for is
Tylenol or aspirin or Advil.
You went on to say that you should actually be drinking.
Well, it's clinically shown. I mean, why does fever? I mean, why does your body temperature rises when you have an infection? It's a defense mechanism. It's trying to kill bacteria and viruses. By suppressing it, you're actually doing the opposite. So it's actually clinically suggested, as long as your body temperature doesn't go above 40, 41, it's okay. It's a defense mechanism. It's good for you. But I don't know anybody, including myself, even though I know it, that does that. As soon as I get fever, where's the aspirin? Where's Tylenol? Because it's uncomfortable. You feel sick. You don't want to feel sick. But it's a defense mechanism. It's a natural defense mechanism, but we don't like it, so we suppress it. It should be left alone for about 24 hours at least. Yeah, I mean, at least that's based on my knowledge, but maybe somebody will disagree. Camilla, you can ask your dad, see whether he disagrees or agrees with me. Definitely not in pediatrics, though. As soon as they get fever, that's it. We're giving them Tylenol. You know, nobody wants to play with the kid because they have a very small organism, obviously, and the changes happen very fast. It could be 37 today, and in an hour it can be 40. That quickly. Anything else? Yeah.
Proteins that you work with
Yeah.
specific for human?
Yes. Yeah, they're all human versions. Even though I use human cells as well. Not always. In this case, they were humans. Okay, well, if there's nothing else, thank you for your attention. Appreciate it.